So we've solved one of the problems, right? We've got the eggs and sperm up there in 2 dimensions. We're maximizing the contact between the 2 of them. So our next problem is we've got these coral eggs out there, and we've got all these sperm trying to rush to these eggs and fertilize them. Well, it's great if one sperm does that, but it's bad if more than one sperm does that. So if we get polyspermy, so multiple sperm fertilizing an egg is bad, because the eggs, or the larvae that makes, they don't develop in the right way, and essentially it's wasted effort. So the first step, the sperm finding an egg, these eggs often release chemical cues that these sperm can sense, and they can actually track over small distances to the egg. So once these two come in contact, you get the acrosome reaction, so essentially the breaking down of this jelly coat on the egg, which actually allows the sperm to get inside to the genetic material inside the egg. So once that happens, you want to immediately block any other sperm undergoing that acrosome reaction and having a second fertilization of that egg. So what happens in most marine species is you get what's called a fast block to polyspermy. And essentially, before these two fertilize, the sperm has a positive charge on its outside, and the egg essentially has a negative charge on its outside. Well, as soon as this egg gets fertilized, that polarization flips, right? And that egg then has a positive charge running around its outside. The sperm is also positively charged, so it's like putting two positive ends of a magnet together, right? And they repel each other. So essentially, you get the reversal of this polarization of the egg, and you essentially get this magnetic force that helps drive the egg and the sperm away from each other instead of attract them. In mammals, it's a lot different. It's called a slow block, where the actual jelly coat changes shape and structure to prevent the sperm from actually getting inside. But we don't worry about that. This fast block is what we see in most marine species, that membrane depolarization. So, okay, we've got all our eggs in 2 dimensions now, and we understand how to prevent multiple sperms from fertilizing a single egg. So how do you prevent the wrong species sperm from fertilizing their egg, right? Say you're in the Pacific Northwest, and you've got these 3 really closely related urchins. You've got Strongylus centrotus franciscanus, which is this guy here, this big kind of light purple guy. Strongylus centrotus purpuratus, which is this big dark purple guy. And then Strongylus centrotus droybacchiensis, which is this little guy. Right, so they're all very closely related. They've only diverged in species in the past few tens of thousands of years. So how do we actually prevent the sperm from droybacchiensis from fertilizing franciscanus? Well, on the egg of these sperm, or on the surface of these sperm and egg, are proteins called binding proteins. And they're very species specific, right? So that the binding proteins of the sperm from droybacchiensis have to recognize the binding proteins on the surface of the eggs from droybacchiensis for fertilization to actually happen, right? So if we get a mismatch of binding proteins, we get eggs from this guy and sperm from this guy that come together, they're not going to fertilize because they have incompatible surface proteins. And this rapid evolution of these proteins over just a few thousand years have led to the speciation and the ability of gametes not to fertilize across these species. Okay, so those are the main ways that our marine invertebrates, or our species that have broadcast spawning, overcome the challenges to external fertilization. All right, the last thing I wanted to say about reproduction before we move on, just say a couple of things about asexual, non-sexual, sometimes called vegetative reproduction. We'll talk about this in more detail in the class when we talk about corals and other species that really rely a lot on asexual reproduction. But remember, we're essentially producing individuals with the same genotype. When a piece of this coral breaks off, falls down, gets stuck in a hole, and keeps growing. Right? So that's vegetative or asexual reproduction, fragmentation. We can take advantage of this asexual reproduction to essentially restore populations. So on all these little ceramic hockey pucks here are little pieces of this staghorn coral, Acropus cervicornis. And a lot of efforts throughout the Caribbean, including here in South Florida, to essentially propagate little pieces of these corals and then outplant them back to natural areas of reef where they used to exist, but now are gone because of disease. There's disease here, rapid tissue loss, and other diseases that have come on this point. So people are taking advantage of this sexual strategy, asexual reproduction, to help restore important species that have lost. So we'll talk about things like vegetative spread, like these salt marsh grasses and their ability to spread throughout the sediment. Just as reproducing asexually through vegetative spread and colonizing these large areas of salt marsh as foundation species. We'll talk more about that when we talk about salt marsh. Okay, so we're going to talk for a little bit, before we run out of time, just quickly about reproductive effort, and then a little bit about larval ecology. So what happens when our egg and sperm actually do get together, make a larvae, then what happens? Where does that larvae go? How does it get there? And how does it find a new place to live? Okay, so when we think about the reproductive effort of a species, you think about how much energy individual species or individuals put into reproduction and how often that input actually happens. And we can essentially break our organisms, both marine and terrestrial and freshwater, up into two different categories. We can think about iteroparous species, or often called iteroparity, or simulparous species, also called simulparity. So when we talk about iteroparous species, we're talking about organisms that produce multiple times throughout their lifespan. It's an iterative process. Right? So things like us, bears, sharks, often big, long-lived animals. Our simulparous species, or animals that have simulparity, are organisms that usually only reproduce once, and then they die. Salmon is a really great example, Pacific salmon. Lots of insects essentially live, reproduce in one massive effort, and then they're toast. All right? So when would each of these strategies of reproduction be favored? What conditions would favor, say, iteroparity?
When the species is able to live a long life